Hello, and to everyone who's listening to yet another edition of the Global Immunotalks. And it is my great pleasure to host today's speaker, Aviv Regev. And as you know, our seminar series ends abruptly. You have probably lots of questions for Aviv after. In order to do so, in order to ask her questions after the seminar, please do so on Twitter. We will upload again the details on how to ask questions after the seminar, but there's no possibility to do it here in the chat room. Again, great pleasure to have Aviv with us. She received, she's a very, very talented scientist, well-known in the field, especially in computational biology. She received her master's from the Tel Aviv University, where she studied biology, computer science, and math. In 2002, she received a PhD in computational biology, also from Tel Aviv University. And then she did something unusual. She didn't do a postdoc, but instead she was invited as to have a fellowship at Harvard, to have a small group starting on her own. And that was in 2003. In 2006 already, she was then recruited to the Broad Institute, and she became a professor also at the MIT. Again, rapidly, 3 years later, she became a Howard Hughes Medical Institute investigator, and she's done a lot of stuff that we all heard about, and we'll talk about it some more. But in 2020, she did something yet again surprising, at least I thought, was to become the head of research and early development at Genentech. So she is no longer an academic, so she's become a commercial, working for a commercial enterprise. There are so many honors, and we don't have time on Global Immunotalks to go through all of the things, but among the top achievements, obviously, is at her tender age that she has become a member of the American National Academy of Sciences and the National Academy of Medicine. Now, Aviv is possibly best known for her study on molecular circuitry that governs the function of mammalian cells, both under steady-state physiological conditions, but also in disease. She's pioneered many experimental and computational methods for the reconstruction of these circuits, including single-cell analysis, genomics, etc. As many of you may know, she's also the co-chair of the organizing committee for the International Human Cell Atlas project, a very, very exciting project. And before I pass over to Aviv, I mean, one thing we do here at Global Immunotalks was we ask our speaker a question that is slightly beyond the science that you're doing. And so the question that we discussed is, Aviv, what's your mentorship style? And was it always like that, or did it evolve over time?
Great. Thanks, Burkhard, for inviting me, and thanks everyone for listening. And mentorship is really near and dear to my heart, so I love getting this question. So maybe I'll start by a loose translation to English of a saying in Hebrew. I'm Israeli originally, so Hebrew is my native language. I've learned from all my teachers, but from my students, I've learned the most. And I think maybe one of the big tenets for me from a mentorship perspective is to realize that it's a two-way street. It's not just that you're mentoring somebody else, but every relationship like that, whoever you mentor is also your mentor, and vice versa. And I think that's how I learn, and that's how I can get to go and do new things that I haven't done before. The other big aspect, I think, around mentorship is that mentorship is very different from other kinds of professional relationships. For example, it's not about managing other people. It's about giving other people advice. And they might take that advice, but they might very well not take it. And sometimes not taking it is more important than taking it. That's the unique thing of mentorship, unlike other relationships, and it's something that I really hold near and dear to my heart. And so that's a second component. The third one that I'll highlight is the ability, especially in our world, in science, the mentor doesn't have the answers. We don't know the answer to a scientific problem when we start working on it. There's no textbook answer. It's not written anywhere. And it's not just the answer that we don't know. We don't even know the path to get there. And that experience of, looking into the abyss and there's nothing there, and yet you still need to make it to the other side, is really essential for scientific research at any stage of your career. And a mentor's role is really to be there, with the right sentence and the right scaffold, to make it possible for the person to do that rather than themselves. They don't know the answer to. That's a really important and fundamental realization. And then finally, I'll say sometimes it's all about being the potted plant, meaning somebody comes to your office, they have something on their mind, they want to talk about it. It can be everything from a research problem to a personal problem. And they sit there and you basically say nothing. They talk through their problem. And by the time they finish talking, they figured out what they're going to do. And they say, thank you. And you say, but I didn't say anything. And sometimes that's the best mentorship of it all. So maybe I'll stop here and turn to the science.
Excellent. I would probably have said, you're welcome, and take ownership of this incredible mentorship. Then, Aviv, that's wonderful. So once again, we are very, very thrilled to have you. And please do share your screen and begin your seminar.
Absolutely. So let me indeed share it. Let me also apologize that I am actually recovering from COVID, and you might hear me cough here and there as I speak more and more. So I'm indeed going to talk today about cells and their properties. Before I start that, I'm going to share with you my disclosures. I am a co-founder and I have equity in two companies called Celsius and Immunitas. And as Burkhard said, until 2020, specifically until July 31st, 2020, I was purely an academic, but I was also on the SAB of Thermo Fisher, Neogene, and Cirrus. And then from August 1st, 2020, I'm an employee of Genentech and I have equity in Roche. So as I think many in this audience know, and as Burkhard a little bit alluded to, single-cell genomics, the ability to profile the molecular content of individual cells, has really shown us that these cellular profiles carry a lot of information on the fundamental properties of cells. And that includes many different things, the types and states of these cells, but also dynamic processes like differentiation and other temporal processes, the programs that the cells run, and even where the cells are physically located in a tissue or in an organ, and how the cells interact with each other in the tissue. Now, if we turn specifically to the context of disease, these give us what I think of as a new vocabulary, or at least a more refined vocabulary, for studies that try to address basic questions in disease, like where might disease genes act, which cells might be disrupted, which programs change in them, how their communications with other cells are affected, and what might be the impact of drugs and resistance mechanisms to treatments. And so today, I'm going to tackle these kinds of questions in the specific context of cancer, where single-cell and spatial genomics can be instrumental to help us decipher the ecosystem of different kinds of cells in the tissue, and they can help us understand key processes in tumorigenesis, drug response, resistance, and metastases. And I know that the title of the seminar series is Immunotalk, and of course, some of the most substantial cells that we have in a tumor, besides the cancer cells, the malignant cells themselves, are different kinds of immune cells. And this is how these two themes are going to come together. So I'm going to illustrate this through a series of stories that tackle with immune-excluded tumors. Then I'm going to turn to the control and functions of immune exclusion programs. And finally, I'm going to talk about how different driver mutations combine to affect both the malignant cell and the tumor microenvironment. If there's time at the end of this talk, I'm going to close with a few upcoming methods and tools that I think are going to be helpful as we try to tackle remaining challenges. So I'm going to start with a patient's tumor. And in this case, the tumor in question is melanoma. As I think many of you know, many patients have derived tremendous benefit from checkpoint inhibitor therapy, where immune cells are unleashed by drugs to go back and attack tumor cells that otherwise they're suppressed in different ways from attacking. Now, many patients derive this benefit, but other patients either have intrinsic resistance, meaning they're resistant to the therapy even before treatment, or they acquire resistance over time after treatment. And one hypothesis in the field for a long time has been that resistance is associated with what we call cold tumors that don't have infiltrating T cells, whereas responsive tumors are hot and they're filled with tumor-infiltrating lymphocytes or TILs. More generally, we often categorize tumors by their portion of T cells and where they are located. So at one end of the spectrum, we have hot inflamed tumors that have a lot of infiltrating T cells. At the other end of the spectrum, we have immune deserts where there's no T cells at all. And in between are what we call immune excluded tumors, where we have T cells, but they're at the periphery of the tumor and they haven't infiltrated into the tumor parenchyma. And so quite a number of years ago now, Livnat Jerby and Ben Izar, when they were postdocs in the lab, each of them has their own lab now, wanted to find out if malignant cells, cancer cells from some patients have properties that make the tumor more likely to exclude T cells and resist immunotherapy. And so for this, they decided to use single-cell RNA-seq to profile the different types of cells in tumors from 31 patients. Half of them were not yet treated, and half of them were after treatment. And all of those after treatment were non-responders. So here we're looking only at the cancer cells, only the malignant cells, although the others were profiled as well. And in the top lot, the cells are colored by the patient, and you can see that the malignant cells from each patient form a separate cluster. But what we're interested in are common features of cancer cells across patients. And so in order to look for these common features of the malignant cells and relate those to another property of the tumor, how much T cell infiltration is there, Livnat developed a computational method. This method looks across many tumors in order to find features in the profile of cancer cells, malignant cells, that correlate with the frequency of T cells in the tumor. That means that the method looked for genes that are expressed in cancer cells, but when they are higher in cancer cells, the T cell levels in the tumor are higher. Or conversely, gene expressed in the cancer cells that were higher in the cancer cells, the T cell levels are lower. And the result of that is on the right. It was a coordinated gene program expressed in the cancer cells that we call the T cell exclusion program. So in this program, genes like MHC class 1 or interferon gamma are low in cancer cells in the cold tumors, and they're high in the hot tumors. And genes like MYC, like MYC targets, CDK4 and CDK targets are high in the cancer cells in the cold tumors, and they're low in hot tumors. So now we first wanted to see whether this program that we inferred basically by a statistical method is present in the cells in situ in the original tumors. And for this, we used a multiplex spatial proteomics method called T-CISIF, developed by Peter Sorger's lab. We profiled with it 13 proteins in sections from 19 of our patients' tumors. And in this way, we can mark the key cell types in the tumor, but also the activity of this specific exclusion program. So to orient you, I'm showing you first just one section. In blue, we see the T cells. CD8 T cells are in light blue. In red, we see malignant cells that repress the exclusion program, so they should promote a hot environment. And in green, we see malignant cells that induce the exclusion program, so they should promote a cold environment. And this is exactly what we see across multiple patient tumors. The red malignant cells are interlaced with the blue TILs, but the green cells, which are also malignant cells, form these big cold patches where we don't see T cells. Okay. So next we wanted to see whether this program is related to the response of the patients to the immunotherapy. So far, we just have a statistical association with features in the tumor, but what might be the clinical outcome? So for this, we studied a separate independent cohort of 112 patients that was collected by Dirk Schadendorf and Eli Van Ellen. And in this cohort, the patient's tumors are biopsied before the patients are treated with checkpoint inhibitor treatments. And then the tumor biopsy is profiled by bulk RNA-seq. In addition, the patients were monitored clinically for their outcome. And what we found was that a higher expression of the exclusion program in these tumors before treatment was predictive of a worse outcome for the patients. So now we wanted to see if there are ways to suppress this program, maybe as a new therapeutic approach. So we went to published RNA profiles from hundreds of drug-treated tumor cell lines. And we looked for drugs that would be more toxic for cells that express the program. And this analysis predicted that CDK4/6 inhibition would suppress this program. Now, if you were observant a few slides ago, you might have noticed that CDK4 and targets of CDK4 and 6 were induced in the exclusion program in malignant cells. So we tested this prediction in cell culture. First, we used cell lines, and then we actually used co-cultures where we grow malignant cells that are isolated directly from tumors, together with T cells, with TILs that were isolated from the same patient tumor, so autologous TILs. And when we treat those with a CDK4/6 inhibitor drug called abemaciclib, it reversed the expression of the program. And then finally, we showed that treating animal models of cancer with a sequential combination, first treating with checkpoint inhibitors and then with a CDK4/6 inhibitor, reduced tumor growth. Now, at this point, we also asked ourselves, is this program really unique to melanoma, or could a similar program actually be found in other kinds of tumors? So we scored the expression of this program in bulk RNA-seq from multiple tumors in TCGA. And we found that cutaneous melanoma, lung adenocarcinoma, and kidney cancer all have low scores. And that's consistent with the benefit of immunotherapy in these tumors. On the other hand, cancer types that are typically not responsive, like uveal melanoma and colon cancer, all have relatively high scores. And the highest score was in a tumor type called synovial sarcoma. Synovial sarcoma is a young adult tumor. It's a very rare tumor type. It's very different than melanoma, where we started from. But we actually pursued this clue in a whole separate study that we did in collaboration with Mario Suva and Nick Ricci. And we ultimately showed that synovial sarcoma cancer cells also express the exclusion program. And in synovial sarcoma, it's also related to T cell suppression and to T cell exclusion. And it turns out that in synovial sarcoma, this program is controlled by the main driver of the tumor, which is a fusion protein called SS18, SSX. Okay. So now all of these analyses I showed you so far, first dissect the tumor into its components part. We dissociate the tumor and we do single cell RNA-seq. And then we assemble them back together, mostly based on known relationships that we're interested in exploring in a very hypothesis-driven way. For example, we know that CD8 T cells and malignant cells interact. Let's look at the relationship between gene expression in malignant cells and the proportion of CD8 T cells. But if you think about this more generally, we expect many different cells in a tissue, and specifically in a tumor, to coordinate their actions, either because they're all responding to a shared signal, or because one of the cells, for example, responds to a cue and it affects the others. Hang on. And in any case, we would expect a signal in the data that would reflect this coordination. And so in order to recover this kind of signal from either single cell data or from spatial genomics data, and I'll show you both examples, Livnat Jerby developed another computational method that we call dialogue. And this method learns what we call cell type specific representations. Now, a cell type specific representation is similar to comparing the expression of one cell type to all other cell types and asking what genes are uniquely expressed in that cell type. We do this all the time, but here there's a twist. And the twist is that the method is going to look for dependencies between cells of different types as it looks for that. It's going to ask, what are genes that characterize this cell type when this other cell type expresses some other genes? And how do you know which two cell types to relate to each other? You look for cells that are either of different types, but present together in the same physical niche, which you can do with spatial data, or are present together in the same sample, which you can even do just with single cell data. So to illustrate this in cancer, we analyzed data from a published melanoma study that was conducted by Gadi Getz and Yohan Cohen's labs, and they profiled just the immune cells of tumors from 48 patients, 31 of them responders and 17 non-responders, all of them by single cell RNA-seq. So we took this published data and we applied dialogue for it. And what dialogue gives us back are these multicellular programs. So one of the multicellular programs that dialogue found is associated with a response to immunotherapy. The induced part of this program is higher in the responders, and it's lower in the non-responders. And the repressed part has the opposite pattern. But unlike before, where the program I showed you was made only of genes that were co-expressed in one cell type, in that case, the malignant cell, here we have different genes in each of 4 different immune cell types. And as a result, this program allows us to connect the expression of macrophage M2-like polarization genes, the repression of ApoE in macrophages, and a dysfunction program expressed in T cells, including of a gene called CXCR6, which is needed for interactions in myeloid cells in the tumor microenvironment, something that we showed in a follow-up study with Anna Anderson. Okay. Now, sometimes we have better than single cell data. We actually have spatial molecular data. And in this case, we can also see how multicellular programs relate in space. So here we applied dialogue to spatial molecular imager data. It's a technology from NanoString, where 980 genes were profiled in situ across 800,000 cells from non-small cell lung cancer tumors from 8 patients. One of the MCPs the dialogue identified spans T cells, macrophages, and fibroblasts. Now, there's T cells, macs, and fibroblasts in various locations in tumors, but those that activate this particular multicellular program are prevalent in the boundary with the tumor, so they likely interact with the malignant cells directly. And this program spans an interferon response that's active across 3 cell types, antigen presentation genes that are active in fibroblasts, and repression of cross-presentation in T cells and in macs. And we think that IL-1 signaling may play a role in coordinating this multicellular program because IL-1B and its agonist IL-1RN are induced in the macrophages, and the receptor IL-1R1 is induced in the fibroblasts at the same time. So to summarize this first part, I showed you how we use single-cell genomics to make a statistical prediction, excuse me, about a pattern in malignant cells that leads to T-cell exclusion. We validated that it predicts the response to immunotherapy in 2 independent cohorts. One of them I showed you, and the other one was done in collaboration with Genevieve Bolland at MGH and supported the same conclusion. And then finally, we used additional computational approaches to predict that it can be reversed by CDK4/6 inhibitors, and we validated this in cell and animal models. Then we showed that it also generalized in other tumor types, especially in synovial sarcoma, possibly in others as well. And finally, I gave you a glimpse to a new method called dialogue, which allows us to recover not just cell type specific programs, but multicellular programs that are coordinated either across samples or in different physical niches. So now we have a program, it's associated with an interesting cellular, physiological, even clinical phenotype. How can we dig deeper into a program like this mechanistically? So in the first study I showed you in melanoma, we just followed 1 pair of genes, CDK4 and 6, out of dozens in one program. And to be honest, we could follow them because conveniently there was also already a small molecule drug called abemaciclib that inhibits them. But if we want to test every gene for its effect, we need to use a genetic screen, and we need to knock out each gene in turn. Also, we would need to check the effect at a program level on a whole gene expression profile. So the readout needs to be an expression profile. And we need to conduct this experiment in co-culture. We want to genetically manipulate a patient's melanoma cancer cells, but we want to grow them together with T cells from the same patient tumor to see the effect. And so Chris Frangi, Johannes Melms, Pratiksha Thakor, and Katie Geiger, in collaboration with Ben Izar, who by then partly did it when he was a postdoc in the lab and partly after he moved to his own lab, decided to pursue this work. And for this, we used Perturb-seq, a method that we developed several years ago, where we combine a pooled CRISPR screen with single-cell RNA-seq as the readout. This allows us to test every gene in the program and give us both the identity of the genetic perturbation and the impact on the profile of the cell. And it can be done in co-culture. So it satisfies most of the things we wanted to solve, but we had one remaining challenge. And this is because many of the best phenotypes we're the most interested in terms of cell phenotypes, are based on cell surface proteins. And some of these canonical proteins are detected very robustly at the protein level, but not always in terms of the RNA that encodes them using single-cell RNA-seq. And so we modified perturb-seq to rely on site-seq, where we stain the perturbed cells by DNA barcoded antibodies so that we can measure the level of these antibodies in the profiling as well. And now with this new setup, we perturbed each of 248 genes from the exclusion program in the patient-derived melanoma cells, and we assessed the impact in two ways. One was using cell viability in co-cultures with T cells to see if the T cells were killing the cancer cells. And the second was on the RNA profiles of the cancer cells. So first, in the cell killing screen, the strongest hits were in genes like beta-2 microglobulin, HLA, and JAK-STAT pathway genes. When we perturb these genes by knockout, we confer resistance to T-cell mediated killing. This is exactly what we would expect, so it's a nice positive control. But even at this point, there were additional genes that conferred resistance when we perturbed them, including CD47, CD58, and CDH19. So we wondered if genes like those are working basically through the same JAK-STAT pathway, or maybe they work through a different one. For this, we turned, to the perturb-seq screens, where we analyzed the profiles of 200,000 patient-derived melanoma cells. We use another computational method that we call MIMOSA in order to fit what we call a regulation matrix. This shows us the impact of each perturbation on the mean expression of each gene across the profiled cells. And now we can start basically teasing apart this matrix that we learned. So first, this is the most prominent portion of this regulation matrix, and it aligns with what we saw in the viability screen. When we perturb JAK-STAT or the interferon gamma receptor, we inhibit a program of MHC and antigen presentation genes, chemokines and cytokines, interferon response genes, and so on. So that explains why it would give us a resistance to T-cell mediated killing. But when we perturb those genes, we also induce the expression of a gene like CD58, which you might remember itself when we knock it out, actually affects T-cell mediated killing. So this is the first clue that it might have an independent mechanism, and I'm going to return to it. So after we had this nice positive control, we looked at the exclusion program genes. So now when we perturb the JAK-STAT pathway genes, this program is increased. But when we perturb each of 22 other genes, including CDK6, MYC, ILF2, DNMT1, and ASCL3, this program is repressed. You remember, CDK6 is the one regulator that we validated before. Now we have 21 additional paths to repress this program. And then finally, we wanted to see how the pieces are wired together. So for this, we looked at the rest of the regulation matrix. On the one hand, we can cluster the perturbations, identifying what we call co-functional modules of perturbations that have a similar impact across the RNA and proteins. And on the other hand, we cluster the RNA and the protein effect, and we can find programs of genes that are co-regulated across the perturbations. And overall, this gives us 6 modules that impact 4 major programs. So for example, in module 1, SOX4, RB1, SP100, and IRF3, amongst other genes, activate a program of cell cycle metabolism, inflammation, and checkpoint genes. SOX4 and RB1 inactivation has been shown before to impact the response to immunotherapy in melanoma or in breast cancer models. These regulators are themselves repressed when we perturb the JAK-STAT pathway. So this particular path is downstream of JAK-STAT. On the other hand, this same program is repressed by module 2 that has genes like CD58 and CXCR4. So now this model basically becomes a map that we can use in order to work out distinct mechanisms that are related to exclusion or resistance to T-cell mediated killing. And to illustrate this, I'm going to highlight just one example, the gene CD58, which I already mentioned before a couple of times, but let me recap. So CD58 was part of the repressed side of the exclusion program in patient tumors. CD58 knockout cells are more resistant to T-cell mediated killing in the viability screen. CD58 is not just activated by the JAK-STAT pathway. In fact, it's induced in the JAK-STAT knockout. The CD58 knockout has a different phenotype than the JAK-STAT knockout in the perturbed site-seq screen, and it didn't affect the expression of antigen presentation genes. And so together, this led us to hypothesize that CD58 loss is a different resistance mechanism. A quick reminder that CD58 is an adhesion protein. It's typically expressed on the surface of antigen-presenting cells, and it binds to CD2 on CD8 T cells or on NK cells. It has come up before in T-cell mediated killing screens, but there is less known about it, partly because there's no known ortholog in the mouse. And so we validated that CD58 knockout melanoma cells are resistant to both T and to NK mediated killing. We also validated the perturbed site-seq result that the CD58 knockout doesn't impact MHC class 1 or 2 expression, but it does induce PD-L1, and this may be one of the mechanisms by which it impacts resistance. And then finally, we went back to the patient data, and we saw that it's downregulated in patient cells after immunotherapy. So this screen that I described to you was large, but it wasn't genome-wide. A genome-wide screen would naively require about 3 million cells to be profiled. It's totally doable. In fact, people have done that already, but it's still a lot. And so Pratiksha, Katie, and Chris now developed a method we call PerturbMe, which is another method in the perturb-seq family. It uses, again, antibody staining, but it does it in two ways. First, we enriched in flow for cells with a phenotype of interest based on a marker or markers using fluorescent labeled antibodies. And then we do perturbed site-seq like before with a DNA barcoded antibody in the same workflow. And we apply it to do an enriched genome-wide screen for regulators of MHC class 1 in melanoma cells. And we profiled 250,000 cells. Now, this might sound like a lot, but with a genome-wide screen, this would mean about 4 cells per perturbation, which are below the limit for a robust statistical signal for us. So usually it would be insufficient. However, after we do the enrichment of PerturbMe, as we hoped, this increased the coverage for impactful perturbations 10 to 40-fold, and now we can relate the perturbations by their effects like before and begin zooming in on specific genes and complexes and pathways. So to summarize this second part, I showed you how we use perturbed site-seq to drill into the T-cell exclusion program. We parsed out this nice, well-established JAK-STAT module, and also 4 other modules that govern 8 programs, and they include the exclusion program. One highlight that I showed you is the distinct role of CD58. And in fact, since this work, Ben has followed up on it, and he has beautiful preprint on the bioarchive that really dissects the mechanism through which this resistance mechanism works. But there's also many other regulators and targets I didn't talk about today. For example, the role of ICAM1, something that we've later on shown together with Marcella Maus in the context of CAR T-cell mediated killing. And finally, I showed you that scale is important and that we can now use PerturbMe to increase scale, not just by profiling more cells, but also by doing it more efficiently. And so up to this point, I focused on a gene regulatory epigenetic view of the tumor, really looking at the gene programs that malignant cells express that might impact their behavior in the context of other cells in the tumor microenvironment, especially T cells. But what about the backdrop of all the driver mutations that are associated with a tumor? In a tumor like melanoma, there is a very large number of mutations. There's an even impressive number of driver mutations amongst them. And understanding retrospectively from tumors which combinations of mutation matters and how they cause cancer, tumorigenesis, resistance, and so on is really challenging. And so the question is, could we use single-cell genomics in an interesting way to help with this? And so quite a number of years ago, Eran Hodis, who started this when he was a grad student, jointly between Levi Garraway and my lab, and ended it up as a clinical fellow years later, actually just this year, Eran decided to engineer a system that would allow us to assess this systematically, starting with healthy primary human melanocytes, the cell of origin for melanoma, and sequentially building 9 genetically distinct isogenic models. As we go through it, we're going to hit 6 common pathways in melanoma. RB, MAP kinase, and telomerase are the first 3. We call these the core mutations, especially because the vast majority of melanomas has all 3. And then we add on top of these either P53, P10, or APC loss of function. And then finally, we add either the P53 or the APC loss of function on top of P10. And I'm going to label the genotypes with these acronyms, CBT, 3PA, And their order also describes the order that we went through this editing tree and gives us all of the genotypes. One of the things we realized early on in this project is that we can build these models without single-cell cloning. And this is because we can rely solely on the growing replicative advantage, the increase in replicative advantage of edited cells compared to non-edited cells or cells that have fewer pathways edited in them. As we hit more and more pathways, the cell get a replicative advantage, and so they take over the culture every time, and that's how we avoid single-cell cloning. Once we hit the 3 core mutations together, we get immortal melanocytes. This only happens after telomerase activation. But now I want to understand what other characteristics these cells might have. And so we turned to characterize the cells molecularly, first profiling them in vitro. Here are each of the cells' profiles, and they're color-coded by the genotype. And you can see that the consecutive mutations also produce an ordered expression progression in this space. And there's a couple of major transition points. For example, the one when you hit the 3 core mutations, when you go from CB in green to CBT in red, you can see how you pass through a narrow expression change. So we wanted to dissect the underlying expression changes. And for this, we learned gene programs. One program can be related to more than one genotype. For example, we have an interferon program that's induced by the first 2 mutations and is shut off thereafter. And we have an EMT program that marks the main transition through the 3 core mutations. And so on. There's a Wnt program, a lanosome program, MICTOR, and phases of the cell cycle. Okay, that was in vitro, but what might happen in vivo? For this, we took the cells from each of the genotypes, and we looked at their tumor phenotypes in NSG mice. So cells that have the 3 core mutations together, these immortalized cells, they give small nodules in 2 to 4 months, and they give small melanomas in 5 months or more. Now we look across all the genotypes, and we see how different combinations of mutations are associated with different phenotypes in vivo. We get primary tumor growth and at least some metastases every time we add a 4th mutation to the core. And then we get pigmentation only with the A or the P3 combinations, and only the CBTPA has this very aggressive disease with wasting. After this, we went and isolated and profiled from the tumors both the malignant cells, which are actually human engineered cells, and the mouse tumor microenvironment cells from the same tumors. And we're going to look at each of them. First, we're looking at the cancer cell. These are human cells, and we color them on the left by the genotype. And you can see that there are substantial differences in the programs across the genotypes, and a really nice agreement between the in vivo and the in vitro programs. But what is even cooler is that the in vivo programs are consistent with the programs that we see in patient tumor cells, directly from patient tumors, even if studies call them by somewhat different names. And even more than that, there's cases where the programs are associated with the same genotype. So for example, both the patient-derived OXPHOS and our in vivo OXPHOS melanocyte program were enriched in tumors that had mutations in the Wnt pathway and that had high levels of pigmentation. Okay, so now finally we turn to the tumor microenvironment and we analyze the programs of the tumor-associated mouse cells. Now these are NSG mice, so there are no B, T, or NK cells, unfortunately, but there are myeloid cells and there's also tumor stroma, such as fibroblasts. And we saw clear distinction in the composition of the tumor microenvironment of tumors with different genotypes and ages. The most dramatic example is with neutrophils. There are nearly 40% of the TME cells in the CBTA and the CBTP3 tumors, but they are a very small minority in the tumors that are derived from any of the other genotypes. And that includes the very close genotype of CBTPA. And this really highlights how important it is to look at different combinations of mutations. Now, if you look at the melanocyte genotype, it also impacted the state of the tumor-infiltrating mouse immune cells. So, for example, there's M1-like macrophages in CBTA and in CBTPA tumors, both of which have a Wnt pathway mutation. And in both cases, they're enriched for a TNF interferon program. So that is something that's affected by the specific Wnt pathway, for example. Also, if we look at neutrophils, even though both CBTA and CBTP3 have large infiltration of neutrophils, and those neutrophils span a range in both cases, the neutrophils in the CBTA tumors have a stronger program, higher expression of a program that's typical for tumor infiltrating neutrophils. And in fact, the CBTA melanocytes express CCL2, which is a known attractant of neutrophils, and that might explain the mechanistic connection. So this really emphasizes the importance of looking at other rich phenotypes. And in fact, we're exploring both H&E stains and multiplex immunofluorescence with our genotypes to better understand these and other features. Just as an initial glimpse into this, we trained the deep neural network classifier on histology sections from the tumors to predict the genotype from histology, and we got excellent performance of the tumors in NSG mice. We even tried to use the classifier on our melanoma models and apply it to histology sections from TCGA, and you can see that for genetics like APC or P53 loss of function, the classifier was not successful. But for predicting expression of related programs, there was some signal. Just to clarify, this is data trained wholly on the mouse data and then applied to patient tumors. So to summarize this third part, we built an engineered model of melanoma-relevant mutations, in different combinations on the background of primary human melanocytes. These have different phenotypes. They hit immortality with the first 3 mutations, and then they accrue in aggression as more and more pathways are hit. Single-cell RNA-seq connects the mutations to changes in cell intrinsic programs in vitro and in vivo, and it matches what we see in patient tumors. Also, the mutations impact the composition and gene programs of the microenvironment and the histology. And we're now expanding this approach to other mutation combinations, other readouts, and other perturbations. So in the remaining minutes, which I do have, I believe, I would like to give you a little bit of a quick glimpse into some upcoming methods that I think are going to be helpful in further dissecting tumor ecosystems and actually other tissue circuits as well, just like single cell and spatial genomics were quite beneficial. And in particular, I want to focus on the challenge of mapping between levels in biology. For example, how do we relate between a cell or a tissue image and a molecular profile? Usually, in order to do this, we expect to have to measure 2 levels simultaneously. For example, measure molecules in situ in a cell or a tissue, just like I showed you. And often, this need for measurement means some hard trade-offs. And one of the biggest trade-offs that we have is between analyzing live cells and tissues and doing comprehensive but very destructive profiling. And so the question is there a way for us somehow to have both using methods like Raman microscopy? So what is Raman microscopy? Raman microscopy is a label-free, non-destructive measurement of vibrational energy levels of molecules, and it is done at subcellular spatial resolution in live cells. So essentially, Raman is measuring molecules through these vibrational energies, and it is doing it in live cells. But it does have a drawback. It's not yet interpretable in molecular genomic terms. I can't tell you which molecule it is, like which RNA or which protein. And so Koseki Kobayashi, who's a postdoc in the lab, developed a method we call Raman to RNA, or R2R, where he trains a model to infer single-cell RNA profiles from Raman hyperspectral images. To train this model, Koseki is going to use single-molecule FISH data of selected markers as an intermediate. Then the trained model is going to generate single-cell expression profiles from label-free live Raman images without doing additional single-molecule FISH experiments. So to test this idea, we focused on a mouse iPSC reprogramming system where we had ample previous single-cell RNA-seq. Koseki collected Raman imaging for 500 cells in each time point. And then he fixed that culture, and he collected matching single molecule fish for 9 anchor genes. Here's the first striking result. It's just an embedding of the Raman spectra along the time course, colored by the time point. And you can see that there's this encoded temporal dynamics and branching. And if you've ever seen single-cell RNA-seq data, it usually looks something like that. And in fact, here is single-cell RNA-seq data of the same time span of the same biological system. You can see the similarity and the mirroring. So after training R2R on these data, the inferred and the measured single-cell RNA-seq matched in pseudobulk for each cell subset, and when we projected them together. And so we can make R2R work. It's important to know we really needed the Raman. Doing it with bright field was not successful. So here we still needed single molecule fish as a crutch for the training. But we think that in the future, we should be able to do this without any single molecule fish anchors, just Raman and single cell RNA-seq. And why is that? The reason for this is exactly what we did in a related problem of connecting H&E stains, where we have spatial but no molecular information, and single cell RNA-seq, where we have molecular but no spatial information. So Charles Comiter, when he was an undergrad in the lab, developed a method that we called SHAF, where we trained 2 coupled adversarial autoencoders using H&E stains and single cell RNA-seq from matched set of, that's matched in the same set of tumors. And so one section for each tumor was stained by H&E, and the other was profiled by single nucleus RNA-seq. And once we train the model, we serve it an H&E stain, and that generates, the model then generates new single-cell RNA-seq profiles, and in the generative process, it actually maps them at the level of individual tiles in the H&E. So we tested this in 2 tumor types, metastatic breast cancer and small cell lung cancer. The generated single-cell RNA-seq on completely withheld tumors led to gene correlation structures and expression distributions and even new maps that were significantly similar to the real single nucleus RNA-seq of these tumors, unlike a null model. And in the case of MBC, we also had MERFISH data. That's a multiplex FISH method on adjacent sections. And we got a good correlation between overlaid spatial tiles that were inferred by SHAF compared to the ones measured by MERFISH, as well as to expert pathologist annotations. Here we show it for one tumor, and here it is for another. The next level of mapping that we're tackling is understanding causality, or how things combine within and between levels to cause the phenotype that we observe. Like, how do you go from the genes, and especially the gene combinations in our genome, mutated or otherwise, and obtain the variety of phenotypes at different levels of organization. Now, I already showed you an example with a few hundred genes that we studied by perturbed site-seq, 1 gene at a time. But I also showed you an example in melanoma, where we had combinations of up to 6 mutations, and it's the combination of mutations that matter. To illustrate this idea around combinations with another example, here's a recent perturbed-seq study that we did with Sigal Kadoshin's lab to look at the contributions of different subunits in the different variants of the BAF complex, a major complex that's involved in many different biological functions, including immune functions and in cancer. So when we perturb each of 28 individual subunits, just knocking them out one at a time, we see that subunits of the same complex, perturbing subunits of the same complex, lead to related phenotypes at the gene, at the cell state, and at the chromatin state phenotype. Now we went and perturbed pairs of subunits in different kinds of constellations. We perturbed parallel pairs, either in the same complex, but mutually exclusive. Okay, so different parallel families, or in different complexes, but mutually exclusive, or in different complexes, but coexisting. And we also perturbed pairs of non-paralogs in the same subcomplex, and pairs of subcomplexes with specific subunits, but with a core that's shared across all the complexes. I don't expect you to track all the different combinations that quickly, but just to know that there were many different ones. And overall, we found that when genetic interactions are present, meaning the whole is different than the sum of its parts, they are positive. And they occur mostly between parallels and within the same complex, like SMARC1 and SMARC2. On the other hand, there are few intercomplex interactions. When we perturb between subcomplexes, it leads mostly to additive effects. And so this means that the system is quite modular and that this kind of modularity is important. Well, why is that? Because while we can technically assay combinations, they remain a major challenge. The number of possible hypothetical interactions to test experimentally far exceeds the number of cells that we would ever be able to profile, and in many cases exceeds the number of cells that actually exist. And so we cannot just test every possibility. So one would look for some way of shrinking this problem and still getting our answer. And for this, we're going to look again at this regulation matrix. Remember how structured it is. The perturbed genes form these co-functional modules, and the responding genes form co-regulated programs. Maybe there's ways that we can use this structure to make more efficient experiments or to make predictions based on the experiments we did about perturbations that we didn't do. And so several years ago, Brian Cleary, when he was a student in the lab, we suggested that this should be possible, either by no longer measuring, say, individual genes and individual perturbations, but their compositions, or by filling in for experiments that we have never performed. And what do I mean by this? There are three ways that we work on to do this. One is to sum the impact of perturbations instead of individual ones, for example, by profiling little groups of cells and then computationally decompressing the single knockout effects. Another is to have multiple perturbations within one cell and to try to decompress the effects of singles or any lower order combinations from them. And a third approach is to have a mixture of single and multiple perturbations and to try and predict other combinations that were not measured in the experiment. We actually work on each of these three strategies. Each of them works in a different way, and I will be happy to share more about this in the future. And so I told you today about how profiling single cells in tumors helps us understand immune-excluded tumors in melanoma and in synovial sarcoma, helps us dissect the regulation of these programs, and also helps us study the impact of combinations of driver mutations, not just on malignant cells, but also on their microenvironment. I also told you about different upcoming tools to recover multicellular programs, to do more efficient perturb me experiments, to relate between molecules and images, to do that in live cells, or to do it in tumor sections, and also to begin to tackle the huge combinatorial space of perturbations. I hope that I also convinced you that this emerging power of single-cell genomics, we can use reference human atlases, disease atlases, perturb-seq screens, and they help us impact many aspects of understanding and treatment of disease. This kind of power and success came from the nexus of three levers or catalysts that we've had in the past decade. One is human biology, studying biology directly in humans so that human is the model organism, like we have shown you in melanoma and other tumor types. Lab methods that are both high resolution and massive scale, like drop-seq and perturb-seq and spatial profiling. And then finally, the power of algorithms to iterate with such data to yield biological insights, make predictions on experiments that we did not do, and imagine new kinds of experiments that might be powered by math. And I do believe that putting these together gives us a roadmap to better decipher human biology and human disease. But if we want to use this map and really deliver for patients, there's a missing piece. And this has really been my motivation to the change that Burkhard described, which is moving to Genentech, where we combine these three things with the ability to develop new therapies, whatever they might be. And this last piece is, of course, very personal for me. I'm very excited about what the future holds for us by bringing them all together. And so I want to thank the people who did the work and all of you for listening. I highlighted each individual lead and collaborator, but I do want to extend a special thank you to Orit Rosenblatt-Rosen, my partner in crime in each and every one of the projects I've described and the head of our cell and tissue genomics department. in Genentech. And above all, I want to thank patients and their families for their participation in research. Thank you very much.
Aviv, thank you very much. That was a fantastic presentation. I enjoyed every moment of it. I thought it was great fun. And I'm sure there will be plenty of questions coming from the audience. And Fazia, can you put in the slide that shows people how to place their questions on Twitter as a reminder? Okay, here we go. So this, so once again, on behalf of Global Immunotalks, this is if you have questions, and I'm sure there are some questions for you, then you should go to this Twitter page, ask questions for Dr. Aviv Regev here, and then Aviv will try to answer those questions. She assured us she will try to answer the questions whenever she finds the time to do so. Please make free use of this resource. And once again, on behalf of Global Immunotalks and everybody who listened here today and people who will be listening to you in the future on YouTube, Aviv, thank you so much for being here. And thanks so much for sharing your exciting view on what you're doing. Thank you very much.
Thank you.